I'm going to talk today about some of the research we've started here in my lab at MSU, and the goal is to help move toward creating joints that look like this. And so, unfortunately, many people suffer from osteoarthritis, and this is a picture from an arthroscopy of an osteoarthritic knee joint. In this joint, the cartilage has actually been deteriorated down so that this yellow tissue is the subchondral, the bone underneath the cartilage, and it's incredibly inflamed, shown by the presence of this red synovium. If we were to cut arthritic knees into sections, they would look like this, and there would be lesions in the cartilage accompanied by the loss of this red staining. And so this is what, unfortunately, many of our knees look like, and we'd like to be able to put ourselves in a position to move toward knees that look like this, where there are a smooth, healthy surface of the cartilage with lots of this red staining, as well as with having a smooth, white, glistening type of cartilage surface. I think this is what we all want our joints to look like, and hopefully we will be in a position in the next decade or so to move there. And so I'd like to start by thanking a lot of the people who have really made this research possible, particularly my team of students. I've been fortunate to have a wonderful array of undergraduate and graduate students here at MSU to help me get my lab started. I feel like one of the things we enjoy at MSU is incredible institutional support, and so I'd like to thank Dean Gunnick, all of the senior faculty in my department, lots of other collaborators across campus who have really made this work possible. I've got a few external collaborators who have been great to work with, and then most important, I'd like to thank the nominating committee and the family of Phil Capriva, class of 1957, for supporting this seminar series that lets us be here today. And so the motivation behind this work is that at least 50 million Americans have some form of osteoarthritis. And so, again, this is what we all, I think, would like our knees to look like. The cartilage is smooth and glistening. And then, unfortunately, in the end stage of disease here, the cartilage deteriorates and is worn away. And so oftentimes there's bone-on-bone contact, which makes for painful walking, painful joint motion, and these joints become stiff and they don't function as well. This oftentimes leads to a vicious cycle, so to speak, where because patients cannot move as much, they stop exercising and put on weight, which increases the loading on the joint. And so we're going to talk a lot about this cartoon here today. And this is just a cartoon schematic of a knee joint. So imagine a knee joint, and the cartilage is the thin gray tissue on the ends of the bones, and the bones here I've shown in black. And so we're going to be thinking about this a lot today. And the motivation is that a lot of people have this disease in the United States. And so one reason I think osteoarthritis research is of increasing importance is that we have an aging population in the United States. And here on the top, we're looking at the prevalence of adults with this disease as a function of age. And unfortunately, it's worse for women than men, so women represented in blue. And generally by the age of about 65, almost 50% of people have some signs by x-rays of osteoarthritis. And this is what's generally responsible for our joints becoming stiff overnight as we age. And so with an aging population, I think it's important that we try and understand this disease so that we can hopefully begin treating it. And so cartilage, this tissue here, it's fundamentally, I would say, a mechanical tissue. And so cartilage enables smooth articulation between our joints so that we can walk around with relative efficiency. And there's a lot of good data out there that show that the cells in the cartilage can respond biologically to mechanical loading. So use it or lose it, I think, is a fairly good heuristic for thinking about how to maintain our joints and maintain their health. I'd like to highlight one study here which looked at patients who were in their 9th decade of life and did not have any signs of osteoarthritis. And they compared those patients to a variety of other patients across many different backgrounds. And what they found was that patients who had a history of heavy mechanical work were about 7 times more likely not to have this disease in their 9th decade of life. And so this is, I think, it's supporting evidence to suggest that we need to move our joints if we want to maintain joint health. And so use it or lose it, I think. And in contrast, though, too much loading can sometimes be dangerous. And so I'm fortunate to work with Tom Youngst here in mechanical engineering. And Tom was kind enough to share a picture of his x-rays for me. And Tom, unfortunately, has bone-on-bone contact in his knees. And he is toward the end stage of disease where he's probably going to need a joint replacement here soon. And he's done a good job of using all of the expected therapies, but unfortunately they're no longer working. And like many patients, this is what happens. They listen to everything their doctor says, and ultimately these patients need a joint replacement. But Tom Youngst is not your average patient. So he has a history of aggressive extreme skiing, which probably supplied some higher than average mechanical loads to his joints. And so there are some instances where too much loading could potentially be harmful. And I think it's important to realize that there's always a balance, and maintaining a healthy amount of loading is what appears to be healthy for your joints. I wanted to talk also about the current state of the art for treatment options for osteoarthritis. And unfortunately, there is not a lot of information on this slide. And that's because there really are not that many things that we can do for patients with osteoarthritis. So everyone can benefit from regular exercise and maintaining healthy weight. People with mild to moderate disease can take anti-inflammatories, Tylenol, ibuprofen. They can begin to have some steroid injections. As the disease gets worse, there is a subpopulation of patients, we're not sure why, but certain patients seem to respond to injections of lubricant, which will last maybe for 6 weeks and help minimize the pain and improve the function for about 6 weeks. And then more steroid injections. You don't want to have too many steroid injections because they really affect how your bone functions, and they can result in substantial weakening of your bone if you have too many of them. And then continuing with pain management. Finally, as you get to the severe stage of the disease, unloading braces. So Tom Youngst, who we just mentioned, had unloading braces that worked for, I believe, maybe 18 months. And then ultimately, most patients end up at this stage of joint replacement, where this is a procedure that requires a highly trained orthopedic surgeon who basically saws off the ends of your bones and implants metal and plastic pieces that, in the best circumstances, last for about 20 years. And it's great that they last 20 years. The problem is that many patients are not eligible for the major surgery that this is. It generally costs $50,000 to $75,000 per joint replacement, so it's an expensive procedure. And finally, they can only do it generally maybe 2 or 3 times if you're exceedingly lucky. So we're noticing now a lot of patients who have had knee damage earlier on in their lives, and they might need their first knee replacement in their late 40s or early 50s. And if they only get 10, 15 years out of a replacement, by the time they're looking at the end of their life, it's really, there aren't many options left. And so I think this is what the current state of the art is, and there's a lot of opportunities for us to improve here in terms of how we treat patients with osteoarthritis, OA. And so one of the things that I think we can do to improve is that we can have better methods to diagnose the disease earlier. So right now, the gold standard in the US is x-rays to diagnose the disease, and that has some limitations because it's taking basically a 2-dimensional image of a 3-dimensional joint. And so if we have better imaging methods and maybe some blood or other quantifiable markers, we might be able to diagnose the disease a little bit earlier. And that would be great because if we diagnose it earlier, we might have a better shot at treating it. I think we need improved methods for getting drugs to cartilage, what's actually deteriorating. And then most important, and what we're going to spend most time on today, is an improved understanding of how our joints work, the interactions between the biomechanics and the biology. I think if we really drill down into the details of this, it will help us come up with new targets for drugs that can help improve patient care. And so the motivation behind the work in our lab is that every day, all the time, every step we take, we are applying mechanical energy to our joints. And so people have measured contact pressures in excess of 2,600 psi in hip joints, so extremely high. This is a very harsh mechanical environment. Those are very high loads. The friction coefficient of healthy cartilage-on-cartilage lubrication is about 0.001. And it's only really been within the last decade or so that man has been able to make materials that have such low friction coefficients. And really, we can't get there consistently. And so cartilage-on-cartilage is an incredible lubrication system. So this is a trace here of the pressure in an instrumented hip joint as a function of time. And so generally we take about 1 step per second, and we can see that with each step we're applying pressure to this hip joint. Furthermore, there can be faster loads here. So in the case where a patient, for instance, does a single forward hop, there can be loading transients that are as short as, say, 7 milliseconds. And so there's a lot of fast mechanical loading that happens to our joints. And what happens is that mechanical loading is transmitted into compressions of the cartilage. So this is data from an MRI study out of Duke where they looked at changes in cartilage thickness before and after activity. And what they saw is that after activity, it could be expected that cartilage is compressed by about 10% that's the upper range of healthy cartilage compression. And so cartilage is compressed, and this is part of the story. The other part of the story is that cartilage is a very specialized material. And so from a mechanical engineering perspective, cartilage is actually considered a viscoelastic material. And what that means is that each time there's a cycle of loading in cartilage, that there is energy that is dissipated within the cartilage. And so our long-term goal in our lab is to actually develop drugs to repair cartilage synergistically with this normal mechanical loading. We want to see if we can maybe harness some of that energy dissipation in the cartilage itself to help the tissue heal itself. In the short term, so the next 5 to 10 years, what we're hoping to do is to find new drug targets that can respond to cartilage loading. And so the research question we're going to talk about today is, can cartilage cells change their energy metabolism, how they use energy in response to mechanical loading? And to answer that, we're first going to I'm first going to describe some of the methods that we use for these studies so that you guys can really fully understand and appreciate the data. And so all of the data that I'm going to show today were really generated with assistance from a collaboration with Bridger Orthopedics. Dr. Martin Gelbke has been an incredible asset in terms of providing the tissues that he would normally discard during a joint replacement to our lab to study. So we get about 125, 150 joints a year from patients undergoing knee and hip replacements just from a single surgeon here in Bozeman. And that also speaks a little bit to the magnitude of the problem. With one surgeon in Bozeman is doing 125, 150 of these a year, there are many of these procedures happening. And so what we want to do with this the first methods we were developing was to see if we could actually compress cartilage cells in our laboratory. And so the first thing we have to do that is we know that we can get the tissue from the operating room and that we can purify the cells. And so I'm not going to talk that much about that part of the study because we're able to do that, and it's something that's been pretty well described. However, what we needed to do is we needed to encapsulate the cells in a three-dimensional environment so that we could actually compress them. So we couldn't just compress the cells on a flat petri dish as they're normally grown on, because you can't really compress something that's flat that well. And so we wanted to see if we could use a gel, which is shown here in black, and we're using a gel called agarose. We're using that because previous studies had documented that cartilage cells could survive well and thrive in agarose. And here, what we're looking at, all of these little green dots are living cartilage cells inside this gel called agarose. And so what we needed to do was to experimentally model what's happening inside real cartilage. And so this is an image of a real piece of cartilage. And this ring here is called the pericellular matrix. So this hole is where there used to be a cartilage cell that's called a chondrocyte. And so what we need to do in order to model this system in the lab is we need to model the stiffness of this thin ring that surrounds these cells. And so this stiffness has a value of about 25 to 200. And one of the challenges in the literature at this point was that most previous studies were using gels that had a stiffness of maybe 5 or 10. And so it was tough to interpret the results because we didn't know what was happening. And so we started we had built a machine where we could determine the stiffness of these gels. And the most relevant stiffness measure is probably shown in red here, the equilibrium stiffness in these experiments. And so we looked over a range of about 3 to 5% agarose concentration. And this dashed line shows that 25 kilopascal limit. And what we see is that above a concentration of about 4%, we end up with a stiffness that is in the range, the bottom end of the range, but the range of the physiological loading, so what we see in cartilage. And so we wanted to see if we could keep cells alive in cartilage. And so we did this experiment where we looked across a range again of of the gel concentrations. And we did an experiment called a live-dead experiment, where the live cells appear to be green and dead cells appear red when you apply certain staining techniques to them. And we looked at different locations within, say, this gray box is a cross-section of one of our gels, so each symbol up here represents a different spatial position in the gels. And we found that no matter what concentration really we used or where we were in the gels, we were generally getting a live cell rate of greater than about 95%. And so we think we can keep the cells happy inside these gels, and we think that it's a good system for applying compression in the laboratory. And so then we wanted to see and ask the question if we could actually compress cells in this gel with this high stiffness matrix. And so what we did was we had a capstone senior design team here. So these two seniors, Kyle Gunnarsson and Jeff Rooney at the time, they had a senior design project, and they built a machine to interface with the stage of this confocal microscope in the Center for Biofilm Engineering. And so this is a computer model of their design, and this is what it actually looks like. And this motor here would attach, and we could actually compress a sample that sits right in there. And we could do it while we were taking these pictures on a fancy high-resolution microscope, which was great. And so we needed a way to check to see if our gels were going to respond uniformly, that is, if the same compression was going to be applied in the same place in all of the gels. And so we used some small beads that are normally used to calibrate an instrument called a flow cytometer, and we used them as fiducial markers to measure the motion of the gel. And so we did experiments where we would take pictures while we were compressing the gels, and we would track the motion of each of these individual fluorescent beads. And from that, we were able to calculate the mechanical strain. And so we're looking at a couple of different positions here, 6 positions all around the gels, and on a strain scale from 0 to 15% when we're applying a nominal strain that's the same across all of them. And we see that actually generally, looking at these histograms, they're fairly narrow, suggesting that we have fairly uniform strains. The coefficient of variation, the number used to describe that variability, was 6.5%, indicating that it's more than probably about 93.5% homogeneous strain fields across these samples. And so we think not only do we have a gel system that keeps the cells alive, we think we can apply about the same mechanical compression to them. So we think we have a fairly high fidelity system to look at mechanics in these cartilage cells. And so we then did this experiment because we wanted to see if the cells were actually being compressed when we put them in these gels and compressed the gel, because we didn't know if the gel was stiffer or less stiff than the actual cells. And so here on the left, we're looking at samples that were not compressed, and we have the high stiffness, 4.5% agarose on the top, the low stiffness, 2% agarose on the bottom. And each of these rings surrounding these insets is a circle. And so in the uncompressed case, all of the cellular cross-sections appear circular, and that we can quantify that with a ratio of the width to the height. After we apply a 10% compressive strain, we can then look at how these cross-sections might change. And when we're doing that, we can see that in the 4.5% case, this high stiffness agarose, the cells become ellipses in their cross-sections. And when we quantify that, we see that we get a ratio of the width to height greater than 1 that is statistically significant. In contrast, previous studies had used a lot of this 2% agarose, and in 2% agarose, this ratio of the width to height was barely even statistically significant. And so these circular cross-sections resulted even when the overall gel was deformed by 10%, suggesting that maybe 2% agarose is about as stiff as the chondrocytes themselves, and it couldn't actually be used to, if you wanted to compress the cells in a laboratory environment. And so we're happy that we've now developed the system. We know we can keep cells alive, we can apply fairly uniform mechanical compressions to them, and we can now actually compress them. So the next question that we wanted to ask was, can we measure biochemical changes after compression? And so to do that, we are fortunate to be here at MSU, where we have an excellent mass spectrometry core facility. And these facilities that we have here are not really available at every university. And so it's something that we're really fortunate to have. And so what we can do is that we can use an experimental technique called Metabolomics. And what this does is that it quantifies the levels of small molecules that cells produce in biological samples. And so generally, the central dogma of molecular biology is that DNA is transcribed into mRNA, which is then translated into protein. And importantly, at each of these steps, the cellular function can be characterized by a lot of the small molecule cofactors that are helping these processes happen. And so we're going to use this technique of metabolomics to profile how the cell is changing its function in response to compression. And before we did that, we wanted to make sure that, for instance, if we just had a gel with no cells, we didn't detect very many molecules. And so these are plots called mass spectra, and each line on the plot represents how much of a specific molecule we've detected. And so when we do a gel with no cells in it, we detect around 10 molecules. However, when we do a gel with cells in it, so in the blue here, these are agarose gels with cells that have had cells encapsulated within them, we generally detect a couple of thousand of molecules. And so that's wonderful because it gives us a fairly broad measure of how the cell is responding to those specific conditions. And so we next did an experiment where we encapsulated these cells for about 3 days in this high stiffness agarose, and we then compressed them just for 0 to 30 minutes, and we began to look at these metabolomic profiles. And the first result that we're going to present here is depicted here, and this is a type of plot called a heat map, where the color describes how much of a particular molecule is there. And so a red color means that there's a lot of a particular molecule there, and the blue color means that there's not very much of a molecule there. Each row is at a specific experimental condition. So the control of 0 minutes loading is on the top here, 15 minutes of loading is in the middle, and 30 minutes of loading is here on the bottom. And when we perform some computational analyses to see which of these groups are more similar, what we see is that the cluster happens between the two groups that are loaded. So loaded groups are more similar to one another than unloaded groups with 0 minutes of compression. And so there's this idea that the compression can synchronize the cellular behavior that we're seeing in our data. When we do another type of statistical analysis on this data, called a principal components analysis, this seeks to segment sources of variation amongst different experimental groups. What we see is that we have our uncompressed cells, the 0 minutes in circles. We have our 15-minute compressed in squares and our 30 minutes compressed in triangles. And there is substantial separation between these groups so that if we, for instance, zoom way in on this one, we can see that all the individual samples are clustered together. And what this signifies to us is that compression is actually altering the functional state of these cells. So this is good news for us in that we can compress our cells and we can then see changes that we might be able to affect later on. And now this might be the most profound piece of data that we've generated yet so far. So here we're comparing the number of significantly different metabolites between conditions. So here, for instance, we're looking at 0 minutes, so no loading versus 30 minutes of loading here. And we had 348 metabolites that decreased and 357 metabolites that increased. So there was more of these and less of these. And together, this represents almost half of the total metabolites that we detected in these experiments. And so as someone who's interested in the relationships between mechanics and biology, this is very exciting data for us because it lets us know that potentially we could affect half of what's happening in a cell by applying mechanical loading. And so I think this holds great promise for the future in terms of understanding what we can do with, say, the walking around that we might do on a daily basis to help positively affect our joint health. And so what I talked about in the title is using energy metabolism to potentially help repair cartilage. And so now we're going to ask the question, do energy-related metabolites change after compression? And so in doing that, we have this hypothesis here. And so first let me describe the bottom part of this slide here. So this is, on the inside here, this is a cartoon of a cell, what makes up our bodies. And these arrows represent kind of how our cells derive energy from glucose or sugar. And this is a simplified diagram here, but ultimately glucose can lead to different types of molecules like DNA or protein. And importantly, this inset here, again, reminds us of this ring that surrounds cartilage. So this ring is composed mostly of protein. A few different types of protein, type 6 collagen, a growth factor FGF4, perlecan, some glycoproteins, but there are a lot of proteins that are surrounding this. And so our hypothesis is that dynamic compression causes chondrocytes to alter their central metabolism, that is to change the decisions they're making in terms of how this energy is used to promote protein production to support the health of their immediate environment, this protein-based matrix. And so here is some of that data. So instead of before where we were talking about every single metabolite, the thousands that we detect, here we're focusing on a handful of targeted metabolites. And so again, we have just a rotated version of this graph with 0 minutes, 15 and 30 minutes of compression. And we're looking at the relative intensity, that is how much, again, of each of these molecules we detect. And again, the compressed samples are more similar to one another than they are to the uncompressed sample. And furthermore, when we do this principal components analysis here, what we see is that the uncompressed samples shown in green cluster independently from the compressed samples shown in red and blue. And so this, again, is I think, pretty powerful evidence that these energy-related metabolites are clustering, they're behaving together after compression. So as we walk around in our joints, potentially, we are synchronizing in a determined manner how our chondrocytes, our cartilage cells, are using energy. And we can look at this in a little more detail here. So looking at a few ratios here. So we have glycolysis, part of that energy metabolism, and the Tca cycle, another part of that metabolism. We see interesting changes initially, that we see an initiation of this Tca cycle after 15 minutes, which indicates perhaps that some of those molecules produced in the Tca cycle that can lead to produce protein are geared up, the cells are getting ready to make some of those molecules after 15 minutes of loading. Furthermore, we can look at some other molecules that are precursors to other proteins in the cell. And so 3-phosphoglycerate is a molecule that we see increases with loading time, as well as pyruvate, another molecule that's a precursor to an amino acid. So we see increases in these as we compress our chondrocytes more, implying that potentially the cells are going to want to make more of their proteins by making more of the fundamental building blocks to their proteins. And so now we're going to start talking about some of the material that some of the studies we're just starting to do now. And so the first part are mathematical models of energy metabolism. And so this is really a necessary part of this science, because if we think about this cascade of biochemical reactions here that convert glucose into the precursors for protein and other molecules, what we see is that we really have a lot of these kind of reactions where, for instance, one molecule, in this case 1,3-PG, is converted into 3-PG, which is then converted into 2-PG. And so we need a mathematical model for this, because if we, for instance, as we do in our data, we see an increase in this 3-PG, we don't know if it's because of potentially an increase in the production of the precursor, or if it's because of a decrease in the rate of consumption of the downstream molecule. So we can think about this very much as water flowing through a river. And so we don't know if, for instance, say a dam has broken upstream and there's a lot more metabolite flowing downstream when we observe data like this, or if, for instance, there's been some sort of logjam downstream and we're seeing metabolite build up because the downstream side is blocked. And so using this technique called metabolic flux analysis, we can really begin to answer some of these questions. To phrase this differently, right, we might have example data shown in blue or in red where we see a decrease or an increase in a metabolite level. And we really want to understand why. Is it because we're seeing increases in the upstream production, or maybe changes in how much is being consumed downstream, or both? And so we can answer this using a technique from systems biology called metabolic flux analysis. And it's able to analyze all of these molecules at the same time. And so this is a network diagram of energy metabolism. And so glucose goes into our network here, and it can either be transmitted into energy down this pentose phosphate pathway shown on the top here, or down to a branch called glycolysis and into this Tca cycle here. And one reason we're so interested in this is because some precursors to the amino acids that make up proteins are produced in this network. And so 3-PG is produced there, pyruvate is produced there, and another molecule called alpha-ketoglutarate is produced right there. And so one of the motivations behind this network analysis is so that we can understand how potentially these changes in compression change the production of these molecules, which can help us maybe to identify drug targets by, say, we can increase the rate of this reaction here that's going to result in an increase in the production of this precursor to help make cartilage. We're not going to get too much into the nuts and bolts of this, but we can take this network here and put it into a big matrix here that we can that then is represented by what we're using as the letter S for the Stoichiometric Matrix. And so that describes stoichiometrically how molecules change as they're being processed down this network. And then our experimental data give us either accumulation or depletion of metabolites, which we're calling A here. And so looking at this equation, we know that if we have A and if we have S, we can then solve for V. And that's really the point of this modeling, so that we can solve for this V, which are the reaction fluxes or the reaction rates, which can tell us, for example, what reactions we might want to target with drugs, or potentially how a drug is working in order to modify certain reactions. And so what are we going to do when we finish that? Well, we've been thinking about these studies and building up some of them, and we're going to start looking in some mouse models. And so we've had to build a mouse because the way that we normally look at cartilage health is by taking semi-quantitative measures from these sections of mouse knees. And as an engineer, I've always kind of wondered what something that's semi-quantitative means. Is it kind of like being partially pregnant? I don't think there's something that's semi-quantitative. It's either quantitative or it's not. And so this is an example of one of those techniques where this vertical axis represents how bad the osteoarthritis is. So a score of 10 or 12 means that that mouse in that section had a very severe disease, and a score of 0 or 2 means that the disease was not that severe in that mouse knee. And so this graph here represents almost a year of my life, and I was somewhat disappointed at the end when we didn't have really very good precision in terms of discriminating between, say, the different wild type and knockout mice in the different parts of the knee. And so we wanted to come up with a better way to do that. And so we developed this reporter mouse. And so what we did is we bred some mice so that we could end up with the molecule that fireflies use to produce their light inside the cartilage. And so this is an example of when we put the substrate that fireflies use to produce that light into these mice, we see a lot of the cartilage light up. So there's cartilage in the discs in the tail. There's certainly cartilage in the toes and the ankles, and this is actually where the knee is on these mice. Furthermore, we can generate two types of mice. Mice with either one copy of this luciferase molecule or two copies. And we see that the signal is generally brighter in the mice that have two copies of this. And so we wanted to test this mouse by using, again, another one of our senior design projects, which is a treadmill that we made. And so we would run the mice on a treadmill using a 15-degree incline for about 20 minutes a day. And then we performed some baseline imaging to see what the baseline signal was on the mice. And then we would, in half of our mice, we destabilized their left leg with surgery. And we then kept them running for an additional 2 weeks on the treadmill. And we imaged them during the 2 weeks, and then we did metabolomics at the end, but I'm not going to have time, I think, to talk about that today. And so here are what the data look like. So here on the left, these are the mice that did not have injuries. And if we're looking at the left knees of these mice, the signal is about the same in all cases as we go from day 1 to day 15 of this study. Now, in the destabilized mice, we see changes in this signal in this mouse that was being both run on a treadmill after it had its left knee injured. And if we draw a tiny circle around the mouse joints and add up all the signal inside, we can see that there are some interesting changes that happen. So the right knee of the mice that did not receive the surgery appeared to increase for a little while and then maybe decrease, and that's shown in this bright red. In the dark red, the maroon, there was a period toward the end. In general, the signal was always lower in these mice, and then toward the end, they really started to take a dive and to decrease. And so we think we're going to be able to use this mouse moving forward to have a very quantifiable idea of how much cartilage is left in a joint and the function of that cartilage, as we hopefully are able to test some candidate drugs in these treadmill running and surgical destabilization models. And so we started out with the question of, can cartilage cells change their energy metabolism in response to mechanical loading? And perhaps I've convinced you that the answer to that question is yes. And so going back to what we think is needed to improve treatment for osteoarthritis, really, I think we need new targets for drugs to work. And so with our short-term goal, I think we're in a position now to begin to find new targets for drugs that respond to cartilage loading. If we think about this network of energy metabolism here, one of the things this analysis suggests is it possible to funnel energy flow just to maximize these precursors in the joint? And so this potentially gives us some new targets for drugs that we can use that might help to improve osteoarthritis treatment for patients. And so maybe in the future we will be able to add drugs to build cartilage based on metabolic changes that result from daily cartilage loading to our repertoire of treatment options. So with that, I would like to thank my family who has really helped support me through this research. I grew up in a family of kind of engineers, scientists, and teachers, and I married into a family of great communicators, and I hope some of those communication skills have been useful here today. And again, I'd like to thank the people who did this work, particularly the Kopriva family, for supporting the seminar series and as well as our funding sources. This research would really not be possible without support from MSU as well as various federal funding sources that really drive the basic research forward. And so with that, I can conclude. Thank you guys for listening, and I'm happy I'll do my best to answer any questions you may have.